Okay, Chapter 8, Part 2 is meiosis, and the whole point of meiosis is to make babies. This is not cell reproduction for the point of having other cells. We are going to make babies with meiosis, which means we are going to have to take the total number of chromosomes and divide it in half, because anytime you make a baby, you're going to have a mom and a dad. If you left the full set of chromosomes there, the kid would have twice as many chromosomes and would be a mutant. So meiosis is similar to mitosis in like the pictures, but the whole goal here is to get half the number of genetic material in the egg or the sperm. Okay, so sexual reproduction, and we're talking about the fertilization of an egg with sperm producing an offspring. And here we're talking obviously humans, plants, and the advantage to this is that it shuffles the genetic deck of cards. For asexual reproduction, you're going to have identical clones of the parents. It's easier. You don't have to worry about finding a mate. But the huge disadvantage is that there's no genetic variety. So when we talk about natural selection and survival of the fittest, all that good stuff, there's no variety for that to work on. With sexual reproduction, you really shuffle up the genetic deck of cards. Okay, so if we're talking asexual reproduction, the offspring are genetic copies of the parents and of each other. And again, it's easier because you don't have to find a mate. You can just bud off another offspring. But if the temperature of the lake you're in is getting warmer, there's no variety in the genetics for natural selection to work on. Okay. Sexual reproduction, on the other hand, gives a wide variety of offspring, and the numbers are really staggering. It's in the trillions what one couple can produce so far as variety of offspring. And so you'll never see identical children unless they're identical twins. There's enough variety to shuffle this deck of cards that each kid, even if you had, I don't know, 60 kids, whatever, not two of them would be the same. And so that payoff's really, really big. When we look at sexual reproduction, in biological terms, it's expensive. You have to find a mate. Your species in danger of going extinct. If you don't find a mate, it doesn't work. There's a lot of ATP expenditure trying to do sexual reproduction, but the payoff is the genetic variety. All right, so here's the cells arise from pre-existing cells, same as on the mitosis, and the cell division for reproduction is going to come from an existing cell. So the sperm cells and the egg cells are going to be made from existing cells. Okay, this is a hydra, and a hydra is just a common lake little animal, and it can actually just bud off another hydra. That's asexual reproduction. This is done through mitosis, not meiosis. Here you can see a second little hydra budding off. You can think how terrifying it would be if there were two Mrs. Munn cells and I could just clone myself and run around and get all my work done. But humans obviously don't do this. Simpler animals can do that. That is going to be the same genetic material as the parent. They're going to be identical genetically. Okay. So chromosomes are matched in homologous pairs, and that just means homologous means the same. So they're going to be matched. The same ones are going to be matched together. Somatic cells are body cells. So when we talk about your liver, your brain, anything, those cells are body cells. And then when we talk about your gametes, those are going to be your sex cells. So everything else in your body is a somatic cell, and they contain a specific number of chromosomes depending on the species. I'm assuming you're human. We're human, so we have 46. That means each cell has 23 pairs of homologous chromosomes. You got 23 from your mom, and you have 23 from your dad, and so each cell in you has genetic codes from your mom and your dad. All right, so here's your homologous pairs. And they carry genes for the same characteristics at the same place. So maybe right here you got, maybe this is your mom's gene and this one's from your dad. And right here would be eye color, let's just say. And maybe that's blue eye color from your mom, and over here you have brown eyes from your dad. They're going to match up at the same spot. Those spots are called loci, or a locus singular, but loci for plural. And you can see in the homologous pairs they're gonna match up, and that's really important when we get to crossing over. All right, gametes, right? No longer somatic cells. We're moving on to gametes. Have a single set of chromosomes, right? So each time you reproduce, you have to cut the number of chromosomes in half so that you get an egg with half the chromosomes and you get a sperm with half the chromosomes. They come together to make a complete set of chromosomes. So most of you, your somatic cells, your skin cells, are said to be diploid, and that's just 2 sets of chromosomes. Your eggs and your sperm are half that. They're haploid. They're going to contain half that number, and again, by having just a single set, then when the egg and sperm get together, that baby will become diploid, and we'll again have 2 sets. Okay? So to represent this in a diagram, you have meiosis is going to produce an egg, and it's going to produce a sperm. And each one of these is going to have half the number of chromosomes required to be human. And so by each one having half, when they come together in fertilization, we go back to a full diploid set, which is an adult human, either male or female. And obviously the cycle will continue. Meiosis reduces the number of chromosomes from diploid to haploid. I think I've said that like 20 times in this video so far, but that's really, really important. Meiosis, like mitosis, is preceded by chromosome duplication. So the first thing it does is it duplicates the chromosomes. But in meiosis, we're going to do the cell division twice. So even though we've duplicated the cells, we're going to end up with 4 cells at the end. So we'll have half the amount of chromosomes at the end of it. Okay? The first division, meiosis 1, starts with synapsis, the pairing of homologous chromosomes. All right. Then you get crossing over, and crossing over is huge. Homologous chromosomes will exchange corresponding segments. And we'll get a little bit more into that, but what that means is instead of just shuffling the deck of cards with one chromosome or the other in the egg, you can actually mix up the sister chromatids themselves. You can take a piece of one and a piece of another, and that really shuffles the genetic deck of cards. Okay? Then in meiosis 1, you're going to separate each homologous pair, and that's going to produce 2 daughter cells. So we're only halfway there, and they each have 1 set of chromosomes. So they still have a full set of chromosomes. They're still not gametes yet. We have to go through meiosis 2 and get them down to half. Okay, so meiosis 2 looks the same as mitosis. And at the end of it, we're going to have 4 haploid cells. So the process looks the same, but the end result's quite different. We're going to have 4 sperm, or we're going to produce an egg, either way. So we're going to cut the genes in half here. Okay, so here you go. There is a lovely picture of this on page 138 to 139. I suggest you check it out. I'm going to point out some of the different parts from mitosis, the parts that are a little different. So in metaphase, remember in metaphase we actually pulled apart the sister chromatids for mitosis, and here you're going to see that we simply separate. We just pull this one to the side. We're not actually pulling them apart yet. So the homologous chromosomes separate for meiosis 1, but we don't pull them apart until meiosis 2. So we replicate the DNA, same as in mitosis. We line them up, and this part here, the crossing over, is huge. They literally sit right next to each other, and little pieces will cross over. That's why it's called crossing over. And so on one site, you'll have parts of 2 different chromatins. So that's kind of cool. And then let's go on to Meiosis II. All right, so Meiosis II, the revenge. This looks like mitosis. You can see here that we're actually going to pull the sister chromatids are actually going to separate now, just like they did in mitosis. And you are going to end up with 4 haploid cells. Again, I seriously suggest looking at pages 138 to 139 and studying the diagram there. But it's very similar to mitosis. The important difference is you're ending up with 4 haploid daughter cells. And these are gametes. These are not somatic cells. Okay, check out this diagram on page 140. I kind of like it because you're going to start with a single cell going to mitosis and meiosis. So you can follow how they differ. And you can see that this cell only has 2 chromosomes. It has a big one. Here are the 2 sister chromatids, and it has a small one. One from mommy here, one from daddy here, one from mommy here, one from daddy here. When we take it to mitosis this way, the DNA replicates, okay, lines up in the middle in metaphase, lines up in the middle, and we pull those apart at the centromeres, and we end up with 2 identical, sorry, not identical, but 2 cells with the same number of chromosomes. All right, now let's look at meiosis, because this is really key. These go back together, right? So we have the big ones come back together, and they still duplicate. So we had the 2 in the first cell. Now we have 4. So it's called a tetrad because there's 4 of them. And the small ones come back together, and they form a tetrad there. So we've duplicated, but the key thing here with having them together is now we can do this crossing over. And that crossing over trick is important to shake up the genetic material. And then remember, metaphase 1, we just pull them apart. We just pull these tetrads apart, and the chromosomes are still whole, and it's not until meiosis II that we actually separate them out. But in the end, what we end up with are the 4 half cells. So we have one of the larger chromosomes, either from mom or dad, and one of the small, and then one of the large and one of the small. So we have half the number at the end, and we've shaken up the genetic material. Okay, so the independent orientation of chromosomes in meiosis and random fertilization lead to a variety of offspring. What that means is when all these things are going on in meiosis and the chromosomes line up, they're independent of each other. So it's not always mom's stuff on the left and dad's genetics on the right. It's random which way they orient, and it's going to be random which way we pull them apart. And that will lead, so if you had 23 chromosomes pairs, which is for humans, right, 46, but for the genetics we're going to 23 in the gametes, and you randomly chose that, you'll end up with something like 8 million possible combinations for a couple. And when you throw in crossing over on top of it, you end up with something in the trillions. So again, it's all about genetic variety. Okay, so we're going to randomly arrange some of these chromosomes in pairs in metaphase of meiosis. So let's just follow, these just have 2 sets. So they have one of the big chromosomes, one, all right, from mom, one from dad, one from mom, one from dad. And we've already duplicated them, so they formed a tetrad. We've already duplicated the DNA. We pull them apart, and then we pull them apart again. It could be that you get all of mom's DNA in the gametes, and you get all of dad's DNA in the gamete. But it's equally likely that they're oriented this way. There's no reason they can't be oriented switched up. They're independent of each other. So you can get half mom, half dad, and again, we can shake it up even more than that. Okay. Then we have random fertilization of eggs by sperm, and again, that increases. So we've made our eggs randomly, we've made our sperm randomly, and then it's just random which ones get to be the fertilizing one. So all of this is designed to make sure there is variety in the genetic world. Homologous chromosomes carry different versions of genes. So remember I said earlier you can have the brown eye gene from dad or mom or whatever, the blue eye gene from mom. And so you can have it, they're based at the same loci, but you can have different versions of that gene. So it's an eye color gene, but is it blue or brown? So here we go, we have a nice picture of, you have on this, I don't know, dad's, let's just say, you have brown coat color and you have black eye color. And on the other mate, you had white and pink. So we can divide that up. You can get brown with black, and brown with black, and then white with pink, and white with pink. And then when we cross it over, we can actually mix that up even more. Okay, so crossing over. This is huge. Crossing over, crossing over. It's a way of mixing up the genetic material. So on the previous screen, we had brown with, I don't know, remember, brown with black and pink with white. Well, what if we wanted black with pink or whatever that is, or white with the brown eyes? Well, we can get that by crossing those genes over, by mixing them up. So as these tetrads lay out, they literally cross over, and you have areas of your genes that are long. And one possibility is they're extra long in these areas to further crossing over. The longer it is, the more likely it is to cross over. And when I say cross over, I literally mean, look, cross over and then attached. If this used to be red here and going to red here, it now becomes part of the blue gene. All right, and if this used to be blue here and going to red here, this blue piece cuts off and it attaches to the, mom side of the gene. So it literally takes the genes and mixes mom's and dad's sides up. So that on one piece you have some genetic material, your eye color from mom, and you have some genetic material, your hair color from dad. Okay, so in crossing over, we are going to separate coat color from eye color on these chromatids here. So it's nice, they're kind of far apart. You have coat color here, eye color here, and what happens is when they form these tetrads, when they sit side by side, you can see where you get a little break, and then here it literally crosses over. So this blue piece now binds to the red, and the red piece here binds to the blue. That's called crossing over. And the DNA you get is recombined DNA. So it was combined one way before. We have recombined it, so it's called recombinant DNA, but it just means recombined. Literally, we've taken it and we've stuck this piece on it and this piece on it. So now, when we pull it apart, we can have the coat color sorted independently from the eye color. And again, that way increases genetic variability, that you don't have to have, just because they're on the same chromatid, you don't have to have it always go together. This crossing over allows independent assortment of genes. So super cool. Okay. A karyotype is literally just a picture of your chromosomes. So if you're pregnant and you want to look at the baby's chromosomes, make sure they have the correct number of chromosomes, you can do a karyotype and it'll show you a picture of the baby's chromosome. Okay, the way you get a karyotype is you take a blood sample, white blood sample, and you take out just the white blood sample you want, and what you do is you put a factor on them, a growth factor that makes them go through mitosis. And you try to stop it in the middle of metaphase when you have a bunch of chromosomes lined up. And then you can take, it's a literal picture of it, and you can see over here, obviously in nature they don't line up for you nicely when you get the picture. A computer does that, and you can see chromosomes are numbered largest to smallest, generally speaking. So 1 is the biggest chromosome in the human, 2, 3, 4, 5. And so you can look at the picture and you can determine, are there 46, 22 of what are called autosomes, which are just your regular chromosomes, and then the 2 sex chromosomes, an X and a Y, or an X and an X, depending on if it's a girl. So you can actually see that in picture form. If you end up with an extra copy of chromosome 21, what you end up with is Down syndrome. And being one of the larger, the smaller chromosomes, 21 is one of the larger numbers, it means it's a smaller chromosome, it's not usually fatal. If you have an extra copy of, say, chromosome 1 or 2, you wouldn't even survive in utero. The mother would miscarry quite early on, but chromosome 21 is pretty small, so you can have an extra copy of it and end up with Down syndrome. Okay. It is called trisomy, and that just means 3 chromosomes. And if you have an extra copy of it, you can see the karyotype here. You can actually literally see that there are 3 of the 21st chromosome. What's kind of funny is chromosome 21 is the smallest, and you'll notice 22 is slightly bigger, and they're supposed to be numbered by size. I think that was a little mess up when they were numbering these originally. They couldn't tell that 21 was slightly smaller. Okay. The chance of having a Down syndrome child goes up with maternal age. And the thing with eggs is a woman is born with all the eggs that she's ever going to have, and they just mature. Well, as you get older, the genetic damage occurring to your eggs can cause problems later. As they go through meiosis, you can end up with problems. They don't do it quite as well as the younger eggs. And so you can see that after age like 35, 40, it really spikes. It really goes up quite dramatically. Accidents during meiosis can alter the chromosome number, can give you trisomy 21. And it's a result of something called Nondisjunction. And Nondisjunction is when the homologous pairs don't separate in meiosis 1. Remember, one's supposed to go to one side and one's supposed to go to the other side. Or if the sister chromatids don't separate in meiosis 2. If they both go into the same cell, you're going to end up with an extra copy. And if you're the other cell, you're going to end up with no copy of that. All right, so here's a diagram of Nondisjunction. Here you have the tetrad, and it's supposed to be, remember that the first time in meiosis 1, the whole thing is split. We pull one to one side, one to the other side. Well, if both pairs, this whole tetrad goes into one cell, you're going to end up with both of these having an extra chromosome, and over here they're both going to be missing one. All right, then if it occurs in meiosis 2 over here, so the first split was okay up here, it did this correctly, but now down here, one of them doesn't separate at the centromeres here. You end up with this one having an extra set and this one missing one. And then these two here are normal. Okay, so here you have an egg cell that has one extra chromosome there, and you have a normal sperm cell. Well, the baby is going to have a trisomy. Whatever chromosome this is, you now have 3 of them. So depending on which chromosome it is, that can be fatal early on, or if it's one of the X or Y chromosomes, or like we said, trisomy 21, that baby can survive. Okay. Abnormal numbers of sex chromosomes don't usually affect survival. So the X and Ys, if you have an extra one, you don't die. So you don't, like I said, you don't see trisomy 1 or 2, but you can have an extra X or an extra Y, and you'll have different malfunctions, depending on which one you get, or if you're missing one, and obviously it's going to affect the sexual parts of a human being, but it'll also affect other parts. Those chromosomes do other things. We know, being colorblind and that kind of thing, there's other genes located on those cells, on those chromosomes, as well as the sexual features. Okay. So human sex chromosome abnormalities, there's one called Klinefelter syndrome, and you can see in that the person's going to look male, but they have an extra X. So they should have one X and one Y, and they have an extra X. If you have an X and you have 2 Ys, it's not really a problem. That you'll end up having a normal looking male. And if you end up with 3 X's, you end up with what's a fairly normal looking female. If you end up with just an X and you are missing, you don't have an X or a Y, you look female but you have Turner syndrome, and so you will have not fully developed sexual features. Right. So alterations in chromosome structure can cause birth defects or cancer. So chromosome breakage can lead to rearrangements, and that can produce genetic disorders. So we talked in the other slideshow that if you mess up somatic cells, mitosis, you end up with cancer. But when you're talking about meiosis, you're talking about birth defects, genetic birth defects. Okay, so we've talked before about deletions, duplications, inversions, translocations when we're talking about DNA replication. So when we're talking about meiosis, it's the same thing. You can have a straight deletion. You can have a duplication where instead of 1 copy of that, you end up with 2 copies. You can have an inversion, so where it flips from here to here. Some of these, like an inversion, are usually not going to be catastrophic. You still have a copy of the gene. You can have this reciprocal translocation, and that is like crossing over where it's not supposed to happen. It's a non-homologous chromosome, so you're going to end up missing part of a gene, or you're going to have an extra copy of a gene. In fact, this happens with Down syndrome. In some cases, there isn't actually a full 3rd chromosome 21. There's just an extra piece of it. It's crossed over somehow, and you have a piece of it in there. So you either end up with too much of one, or you don't end up with enough. All right, so this bottom figure here, when we go down to here, this is back to mitosis. We're jumping around a little bit. That's okay. This was all meiosis over here. If you have this chromosome 9 and chromosome 22, if you get this reciprocal translocation, which is kind of like crossing over, but it's not supposed to happen in mitosis, you end up with something called a Philadelphia chromosome, and what you're going to get is cancer in this case. This is an activated cancer gene, and it's common, I guess, in certain leukemias that this has happened. So in mitosis, you can have problems like cancer. In meiosis, you get genetic problems in the baby.